Hi, everybody. Today I'm going to talk about reimagining drug development with AI, specifically as it relates to developing optimal drug combinations for COVID-19, as well as other infectious diseases. And I'll branch out into other examples, including oncology. But it's to really look at how we can harness digital medicine to simultaneously optimize which drugs to combine together at which respective dose, and how that can potentially accelerate the drug development process. I'm currently the director of the N1 Institute for Health, also known as N1, the director of the Institute for Digital Medicine, also known as Wisdom, as well as the head of the Department of Biomedical Engineering at the National University of Singapore. And the work that I'm going to discuss today is the product of really wonderful collaborations with clinicians, engineers, healthcare economists, global health security, surveillance experts, and beyond. I'm going to go ahead and turn off my video so it doesn't block any of the slides and start with the presentation today. So in completely rethinking drug development, we have to think about, and there's a need to move beyond the compounds after they are discovered. But starting with this very important process of discovering leads and promising agents, we look at what has happened during COVID-19 and the amazing work that has resulted in screening to reveal repurposing candidates and really neat assays that have identified other repurposing candidates. It's important to think about this pathway or this roadmap to bringing better therapies to patients. And I'm starting off with this paper that I published several months ago, which looks at artificial intelligence in cancer therapy. And this roadmap really is indeed applicable to other indications as well. But we start off by looking at different segments, ranging from drug discovery to drug development to drug administration. And if we think about each category, we have target identification, lead generation, and optimization. In the development segment, there's preclinical development, how we design combination therapies, and how we match patients to the right therapies, all the way to how we dose the different drugs. These are very different segments, but ones that need to be seamlessly integrated to ensure that we can both accelerate and optimize how we bring therapies to patients. If we think about SARS-CoV-2 and COVID-19 and the amazing work that has led to a collection of repurposing candidates, it's important to note that once these candidates are identified, there's an important need to optimize how they are used in the right combinations. And these remaining segments can help bridge this discovery process with optimizing care downstream. And so I'm going to start off with a cartoon that illustrates how AI and digital medicine can globally optimize drug development. And part of that process is really identifying the optimum from massive drug and dose spaces. So I'm going to start off with an example here. And this is a two-drug combination, drugs 1 and 2. Right? When traditional drug development is implemented, there's often this initial identification of a compound that would be suitable for initial development. And it's often 2 drugs, for example. In some cases, the combinations are comprised of 3 drugs. And as we continue to think further, sometimes there are 4 drug combinations. And when we think about how AI may be employed traditionally for this process, we're often asked about, if we have millions of data points looking at these combinations and how they were given to patients, often at fixed dose, can AI be used to potentially predict which combination will be the best for a specific subset of patients? And using traditional AI with these many data points, it may be able to extrapolate out that the 3-drug combination is the best out of these 3 potential scenarios. And again, it's important to note that in many of these scenarios, these drugs are given at fixed dose that were established based on traditional trial designs. But with AI, we can think bigger. So instead of, say, 4 drugs, let's think about 12 drugs, right? And while 12 is not a very big number, the challenge here is that if we think about 12 drugs and knowing that dosing is important in optimizing combo design, and let's think about trying each drug at 10 different doses, that's actually 1 trillion possible drug combinations. And this huge drug dose space is insurmountable in terms of identifying the best possible combination using traditional screening approaches. Let's pretend that drug 5 is ineffective on its own. In traditional screening, drug 5 may no longer be considered. However, we continue to consider drug 5 because it's entirely possible that while it's ineffective on its own, it may enhance the activity of other drugs when co-delivered to result, in fact, a globally optimal combination, right? And so if we think about small data-based AI and digital medicine for drug development, instead of looking at a small number of combinations at fixed dose with a dramatic reduction in the number of data points needed, if we run multiple drug and dose permutations prospectively using actual experimental assays, this affords us the opportunity to outline the boundaries of what is essentially a massive galaxy of drug and dose permutations. And if we strategically run the right number of experiments with the right drug and dose permutations, what we can then do is globally optimize, essentially crowdsource the disease model to tell us what the best possible drugs and doses are, at least at the in vitro level first, with hundreds of experiments, which could represent as many as 1 trillion possibilities, depending on the number of doses needed to interrogate this huge drug dose space, right? So it's important to note that the AI we're going to talk about today does not use pre-existing data to train algorithms and make predictions. In fact, it prospectively runs experiments to outline this parameter space from which the optimums can be identified. And it's critical to note that these optimums, beyond just mechanism of action, can be based upon unforeseen or unpredictable drug interactions that occur out of this pool of drug candidates. So if we think about this a little bit more, this is a multi-stage process where initially a set of drugs are decided, and using an orthogonal array composite design, these permutations of drug combinations are tested on systems of interest. These can be cell lines, they can be patient samples, even preclinical models. The output is essentially a quantifiable measure of efficacy and safety. This can be cell viability, tumor burden, liver enzymes, and beyond. And through an innovative platform known as the Quadratic Phenotypic Optimization Platform, or QPOP, this assay can then lead to response surface mapping, which tells us different drug interactions that can be harnessed to optimize treatment using a much smaller number of experiments. If we look at this platform in action, again, this is pairing prospective data with optimization. And using multiple myeloma as an example, this is an example of where agnostically to mechanism, et cetera, disease mechanism, from larger pools of therapies, unexpected combinations can arise as being optimal. And so if you look at mitomycin C and decitabine, and if you look at their combination together and looking at each drug individually, drugs that may not be very useful on their own towards a specific disease model, when combined at the right dosing together, can result in unexpected and substantial efficacy. If we go from in vitro to in vivo, you can see that this map, which correlates the different drugs and their doses with output, right? And the output can be based upon efficacy and toxicity. You see a change in the shape of this surface. And because we are in vivo, it shows expectedly that, that different dosing and dosing out of the optimal area can lead to substantial toxicity or drop-offs in efficacy as well. And so again, this is all based off of actual prospective data. And it's really important to note that dosing does in fact need to be re-optimized preclinically to ensure that we're reaching this optimal area here. And if you look at the efficacy, the tumor volumes of this mitomycin C decitabine combination that was derived using this approach and prospectively validated, this is in fact more effective than many of the standard regimens, especially in conditions where there may be refractory disease models being used. Beyond that, and preclinical models here, there can be opportunities for patient drug matching, because using biopsies and the implementation of this approach, we can look at patient-specific reports and recommendations of combinations that clinicians may be able to give clinically for subsequent patient-specific combination design. And what's really neat is with the substantial reduction in the number of experiments needed to optimize these rankings, this can be done sometimes within a few days or even a week to actionably recommend combinations. And this work was published a couple years ago, right? And so what we've seen here, again, is not the use of previous data to make predictions. This is the use of prospective experiments paired with optimization to result in actionable answers by identifying globally optimal combinations from very large parameter spaces. It's really important to note that this is a process that can be continuously run and rerun. And what that means is during the in vitro process, our goal here is to reconcile huge drug dose parameter spaces to pinpoint lead combination candidates. After we go to in vivo, we can do more than take this combination from here and give it to a preclinical model. In fact, we re-optimize the dose by presenting these permutations of different doses to different cohorts of the rodents, from which we can derive better dosing parameters that simultaneously account for efficacy as well as toxicity. This is really an important step because synergy that is identified from this in vitro process, that synergy may not be preserved if we don't properly re-optimize how we dose these combinations at the preclinical stage. And moving forward, there are opportunities to even further refine dosing clinically, and we'll show some examples there. So really, this process is to be re-implemented at each stage of development. And so using infectious diseases as an example, several months ago, we convened a team of engineers, of biology and virology researchers, as well as a team of global health security and surveillance experts and healthcare economists to develop a platform that we call Identify, or optimizing infectious disease combination therapy with AI. And this is really, again, mirroring this prospective implementation of experiments to actionably and rapidly pinpoint combinations for indications like COVID-19. And we were able to collaborate with this multidisciplinary team and come to the realization that, you know, at times when healthcare systems and research operations are strained due to epidemics and pandemics, the economic and rapid development of interventions will become increasingly important. And to have this diverse range of expertise was really helpful as we started off in using Identify for infectious disease applications. I also published a perspective paper on how AI can be leveraged, again, for drug development, not just drug discovery, to ensure that these very promising AI or AI-discovered lead candidates can be used and put into the right combinations effectively for further development. And so looking at SARS-CoV-2, using Identify, we ran an experiment where, again, we looked at 12 candidates that were under clinical validation at the time. This is in the March-April timeframe, ranging from remdesivir to favipiravir, ribavirin, Kaletra, and other therapies. And using our approach, we prospectively interrogated the SARS-CoV-2 patient-derived live virus model in vitro to result in a ranked set of combinations that were arrived at via this prospective experimentation plus optimization. And from this work, we showed that with the 12 drugs that we looked at, a combination of remdesivir plus Kaletra was the optimal combination. It's important to note that before looking at these top combinations, remdesivir alone was shown to be the most effective monotherapy, but there was room for improvement, which I'll get into. Kaletra as a combination was shown to be relatively ineffective, as were other combinations that ultimately were shown to be ineffective clinically as well. It's important to note that we arrived at these results with Identify independently of the clinical studies, and we did not require any data from the clinical trials. But because we are globally optimizing these combinations at clinically relevant dosing parameters to make sure we don't overrepresent the promise of certain compounds, we were able to, again, independently arrive at very similar conclusions to many trials. Now, to find out why, right? Why Kaletra is ineffective on its own, and why remdesivir is moderately effective on its own, but why the combination was quite effective, resulting in virtually complete inhibition, we found experimentally, right, that lopinavir and remdesivir have a very favorable interaction, and we know that ritonavir and lopinavir, which comprise Kaletra, have a known interaction. And the interplay of these three agents results in this very substantial increase in efficacy. We independently also showed that experimentally that lopinavir and remdesivir together results in an increase in efficacy as well. And so again, this is experimentally validated both as a 3-drug combination, but as a 2-drug combination as well, to explain why we see what we see. The nice thing about Identify is that our experiments allow for multiple drug candidates to be considered, and this allows us to query a list of combinations ranked based on top 4 drug, top 3 drug. If we need to remove a drug because of it's not accessible, remove a drug because of patient comorbidity, we can query this list in this database to provide us lists of actionable combinations based upon efficacy. And we can see this list tells us which combinations may be relatively ineffective, which combinations, again, may be relatively ineffective, and how that aligns with clinical results. And then again, we can look for higher rank combinations as potential suggestions to the clinical community. And so what's neat is Identify is pretty rapid. The study was completed in about 2 weeks with some wonderful collaborators. And if need be, we can switch up the different therapies to make new rankings and to try different combinations out for identification. And moving forward, with international partners, we've had multiple clinical study or patient-derived sample studies approved that we from our international partners. And I serve on some committees that have seen these examples of how strong technology developer engagement with the community can lead to positive outcomes and potentially actionable data. We received the support of many wonderful partners to continue this forward, and in the near future, we will be running another study with a set of 12 additional therapies, which we will make available for the community. Now, after combinations are developed, there's important additional work needed. Here's an example of HIV patients, 10 patients undergoing combo therapy. And our AI approach has shown that potential drug reduction can lead to undetectable viral load and the potential importance of reducing long-term side effects from these drugs. Novel combinations have also led to important advances in tuberculosis, where with orders of magnitude reduction in drug development costs and reductions in the number of years needed in clinic, treatment time can be cut nearly in half in patients with these AI-discovered repurposed drug combinations. And this allows us to expand this to other indications as well, and we received wonderful support to help make this happen. And this leads us to examples like oncology, where very recently with our institute, we've established a core that allows us to take different samples and rapidly identify actionable combinations for potential clinician recommendation. We started off with an established bank of samples, and now we have protocols in place that will allow us to isolate patient samples rapidly for subsequent analysis and to potentially design regimens for patients within a few days. Once these combinations are identified, again, what we've seen with the infectious diseases as well, we have opportunities to optimize dosing. Here's a patient with advanced prostate cancer where with a 50% dose reduction, we were able to increase efficacy and then subsequently dynamically dose this patient to achieve a durable response. With another patient, we were able to recently show that even from the calibration process alone, even before finishing out the calibration and the dose suggesting, we were able to see some response from the patient as well as prolonged PFS under this low dose regimen. And this was really enabled by a team from multiple facets on campus, many stakeholders coming together to help make this happen quickly. And again, this is an example where even the calibration alone, even before finishing or acquiring all the data points, there can be potential response from the patient that can lead to favorable outcomes. Of note, it's important to validate these types of studies in larger cohorts. And so we have set up a larger pilot trial for solid cancers. And based upon these findings, we'll determine if we will expand to an even larger trial. To bring things towards a close, I think it's important to note that a core ethos of the institutes that I direct, as well as the department that I head, is that technology alone cannot transform healthcare. It really takes the collaboration of different practice-changing methodologies in policy, economics, behavioral sciences, clinical trial innovation, and beyond. And towards that, we've developed a comic book that looks at how AI can address viruses. And this is available for a free download from the Institute website. And this is really a fun project between clinicians, engineers, behavioral scientists, and our wonderful community. And this book is actually, we've been told, has been helpful for many communities beyond early career learners, but also professionals who are looking to learn a little bit more about how AI can be used to address infectious diseases. So today, again, the big summary of the work that I talked about is that with regards to AI, we don't always need pre-existing data and algorithm training to come up with actionable answers. In this particular case, these AI-discovered interactions between drugs and doses and efficacy and safety can lead to prospective experimentation being paired with optimization to yield actionable insight into different combinations that can be given for infectious diseases, for oncology, and beyond. And with that, I wanted to thank the team that makes this all possible, wonderful collaborators, as well as wonderful support from a community of agencies, and we are deeply grateful for the support. And with that, I wanted to thank you for listening in today. And hopefully today we've offered some insight into how AI and digital medicine can improve drug development. Thank you.